Thanks, everybody. Have you ever looked at a really zoomed in photo on your computer? It just looks like random noise, right? But if we could zoom out and look from a different perspective, we'd see patterns emerge that we can't see up close. What looked like random noise before now makes sense. But I want to introduce you to noise in a very different context, noise inside your cells. In order to show you what I mean, I'm going to have to take you on a trip to a bit of a funny place. Actually, we're going to travel inside my nose. Bear with me here. So we're going to travel from this stage all the way down to the level of individual cells inside my nose. Now, here we are. Look around. Be amazed. The nose is actually a pretty amazing thing. It can sense about 1,000 distinct odors. What you might not realize is that each of those odors are actually detected by specialized cells. So some cells do one odor, some cells do another, etc. As far as we know, this decision of what cell senses what odor, it's actually made completely randomly. It's an example of something that we call noise. But what the heck is noise? Well, obviously in this context, it means something a little bit different. Noise is the variability in behavior that we see among genetically identical cells. You should think of that like identical twins. You know that they have the same genes. You don't expect them to behave the same way, right? Noise is just saying that the cells in your body act that way also. I can look at two cells that are right next to each other inside my nose, and they don't have to behave the same way. But I guess the problem with calling it noise is that it sounds like something bad, right? Or at least something that we don't understand. So that was the big question that I had going into my PhD. Is it really all just random? Or are there patterns that we're missing? Because we're looking from the wrong perspective. But working long term on my nose seemed like a bit of a difficult proposition. I needed something simpler, and I ended up picking bacteria. So let me show you why. We're going to conduct a simple experiment right here. We're just going to put these bacteria on a food source and watch what they do by microscopy. What we'll see is just like those cells in the nose, these bacteria randomly change what they do. So you see that? That's noise. That's genetically identical cells behaving differently. But why? Unfortunately, this experiment isn't a very good way to address that question. The basic problem is that it's completely unreproducible. I'm going to do the same experiment 3 different times, and I'll get 3 completely different answers. If we want to find patterns in noise, it's going to be hard if we're looking at something like that. In fact, if we want to find patterns, what we really want to do is watch cells make the same decision over and over and over and over again, exactly the same conditions. And that's where my collaborators and I enter into this story. We used a technique called microfluidics, which let us engineer thousands of tiny traps in which we can capture individual cells. These traps are basically sensory deprivation chambers for cells. So to the cells inside, nothing around them ever changes, and we can basically just park them in there and watch them indefinitely. The end result looks something like this: thousands of bacteria inside thousands of individual traps. Thousands of decisions made in exactly the same conditions. But we're looking for patterns. So are there any? Well, perhaps from looking at this video, you'd think that it looks a bit random. And that's actually true. At this scale, some of the behavior that we see is just random. These bacteria are randomly switching back and forth between 2 different activities. But what I want to emphasize is that random doesn't have to mean useless. In this case, we think they're actually doing it this way on purpose. Switching randomly like this is just like those cells in the nose. It lets different bacteria specialize in different tasks. It's really just division of labor. But of course, if we look in the right places, our new perspective does reveal a hidden order. And these are my favorite bacteria, because these ones actually coordinate their behavior. Once one bacterium decides to change what it's doing, 8 generations of its progeny are carried along with it. That's like you obeying an order from your great-great-great-great-great-grandmother. Not too bad for bacteria, right? And don't forget that we actually have thousands of these decisions to look at. So these are processes that we can start to understand statistically. I can write down mathematical rules and formulas that explain why these cells behave the way they do. How cells make decisions is actually something that we can understand quantitatively from the bottom up. And maybe you think that's neat, but why is it important? Well, the type of variability that I'm talking about doesn't just explain why some cells appear to change color in a movie. It can address some of our most important questions. It can explain why some bacteria resist our antibiotics, even when the rest of their genetically identical brethren are killed off. It can explain why some cancer cells respond differently to our treatments. The basic lesson that noise is giving us is that those cells outwardly look the same to us. They behave like individuals. Understanding what makes cells different then, rather than what makes them the same, can give us a new perspective on some of our most important problems. Perhaps, if we're lucky, we'll see a pattern in the noise. Thank you.